can be enzymatic degradation, or it can be there are other ways of doing it. So, so maybe 200 bases, 400 bases can complete. And smaller fragment, 200 or 400, depending on the platform. So some platform can sequence 200 bases, some platform can sequence 400 bases. So depending on the platform, 200 or 400 bases, if you clip the entire genome, then you can easily sequence, right? So sequencing 200 bases is very less time consuming, right? Even with the Sanger also, for sequencing 200 bases, it takes around 30 minutes, that's all. Now imagine a scenario. In the Sanger you have 96 well capabilities, right? Maximum 96 wells can be sequenced. Now think that you have 100 of Sanger's machine, and all these are parallelly loaded in all the sequencer. Okay? So each machine, each well is capable of sequencing 200 base pair, which will take 30 minutes. So 96 into 10 machine. How much? 960 well. So 960 into 200 fragments can be sequenced within 30 minutes. So basically, now if you join all these 200 bases, so within 30 minutes, what is the read length you have sequenced? So let's round up. Instead of 960, say thousands of sample, 200 into 1000. So how much? 20 kb. So within 30 minutes, you have sequenced 20 kb. So this is the concept of parallel sequencing, and what is exactly done in NGS. So the entire genome is fragmented into smaller fragments, and smaller fragments are very easy to sequence, and within a very short span of time it can be sequenced. Now these small fragments are loaded in the sample loader. So Illumina has chip, glass chip, so it's called flow cell. We have semiconductor chips. So all these chips has billions of small wells that cannot be seen with naked eyes. Just like a 96 well plate, it has billions of wells, and it is loaded in such a way that each well contains one fragment. So that's how the entire billions of fragments are sequenced at a time. And later on, these are again assembled with the help of bioinformatics tools to generate the contiguous fragment, and from there you can get the entire genomic sequence. So, any doubt? You understood what is parallel sequencing? So in the NGS there are certain terminology which are very important. The first term is called reads. So what are reads? So in the Sanger we call chromatograms reads, right? So this is nothing but the FASTA sequences, right? So in the NGS also, this is the output, this is the sequence output, we call it reads. We will not say here sequence chromatogram, we will simply call it reads. So reads are the nothing but sequence output. Then alignment. Alignment is always necessary, whether you do de novo sequencing or you do any resequencing part. So say for example human genome, it is already sequenced. Now you want to sequence your own patient sample. So in that case, what you will do? After sequencing, you have to align with the reference genome, right? So that is called alignment. So alignment is also very important, because after the alignment is done, now you will be confirmed that whether you are getting true sequence or not, or whether the sequence has covered the entire region of interest. Then assembly. So this is mostly done for de novo sequencing as well as any kind of targeted resequencing, exome sequencing, transcriptome. So those short fragments are assembled. So we all have learned about shotgun sequencing, right? So what is shotgun sequencing? Yeah, tell me. What is shotgun sequencing? Just to understand the assembly, I'm asking. Shotgun please don't hesitate. शॉटगन सीक्वेंसिंग क्या है? Depending on the context, so both ends will have some common. So second में shotgun sequence one at a sequence one fragment. वहाँ पे क्या हो रहा है? एक ही fragment को billions of fragments में convert किया जा रहा है and the sequence sample load as billions of plus. क्या हो रहा है? सेकंड में one at a time one well हो रहा है. यहाँ पे क्या हो रहा है ना? Billions of fragments. तो क्या होगा? वो जो 2 घंटे का काम था ना, वो मतलब आपका हाफ एन आवर में खत्म हो जाएगा. एंड नॉट ओनली 1 KB, मतलब आप उससे 20 KB तक जा सकते हो. एज्यूमिंग दैट पूरा 96 वेल्स आपने फ्रेगमेंट करके लोड किया. So that is parallel sequencing. NGS में वही कॉन्सेप्ट इस्तेमाल होता है. That is called NGS. Now different platforms have different chemistry. So here I will speak about ion tolerance. So you will Illumina. So Illumina is also based on reversible chain termination. So it is a modification of Sanger chemistry. But Illumina uses modified chain termination. Nanopore का अलग सा concept होता है. Ion tolerant का अलग सा concept होता है. So these are about different platforms, different chemistries. लेकिन the main part is that you should understand what is NGS and how it differs from the capillary or Sanger sequencing, and what is parallel sequencing, because NGS works on the basis of parallel sequencing. Okay, so assembly में क्या होता है ना, all these fragments are arranged, and with the help of software those common sequences are searched. So once the common sequence between the fragments are found, these are joined. So similar to shotgun sequencing, this is the same principle where shotgun sequencing is done. Okay, so now I am expecting one question from you, that how am I sure that we will have the common sequence? कॉमन सीक्वेंस कैसा आएगा? किसी को मतलब since it is enzymatically degraded, you might have question in your mind that okay, ये एक particular fashion में break होगा तो common कैसे मिलेगा? ऐसा नहीं है. These are clipped randomly. पूरा 3 billion bases को randomly clip करते हैं. So either way, this way or that way, you will have a common sequence between the fragments. And all these fragments are arranged and lined up, and depending on the overlap in the sequence, all these sequences are aligned. So Praveen will take you, my colleague Praveen will show you the bioinformatics part. Actually, I could have shown you if I had blackboard here or whiteboard. another important terminology you will face, it's called library. So what is library? जैसे बुक्स होता है ना, लाइब्रेरी में क्या होता है ना, कलेक्शन ऑफ बुक्स. तो वैसे जब ये सीक्वेंस होगा ना, सो दिस इज कलेक्शन ऑफ डीएनए फ्रेगमेंट्स जो हमें सीक्वेंस करना है. सो इट्स कॉल्ड लाइब्रेरी. ओके. एंड टेम्पलेट क्या है? लाइब्रेरी मिल गया. उसके बाद स्टेप है टेम्पलेटिंग. सो हर एक इंस्ट्रूमेंट का लाइब्रेरी प्रेप का प्रोसीजर्स डिफर करता है. हर एक इंस्ट्रूमेंट का टेम्पलेट प्रिपरेशन भी अलग होता है. लेकिन व्हाट इज द आइडिया बिहाइंड टेम्पलेटिंग? इट इज द एनरिचमेंट ऑफ डिजायर्ड लाइब्रेरी. जैसे हम लोग सेंगर का क्या करते हैं ना, पीसीआर करते हैं. सो पीसीआर इज द एम्प्लीफिकेशन ऑफ योर टारगेट फ्रेगमेंट, राइट? सो वहाँ पर टेम्पलेटिंग में क्या होता है, जो लाइब्रेरी होता है उसको क्लोनली एम्प्लीफाई किया जाता है एंड दैट बिकम्स योर टेम्पलेट. सीक्वेंस जो होना है, एम्प्लीफायबल लाइब्रेरी है, दैट इज कॉल्ड टेम्पलेट. So template is another important terminology. So why I am discretely showing all this is because in the wet lab part you will come across this terminology. Even from analysis point of view also, it is very important to know what is library, what is templating. So in the run parameter or in the result you may see that this is the percentage of template formation, this is the percentage of library. So you should know what is library, what is templating. So why we amplify the library? To get better signal. So that's why amplification is necessary. So in NGS we call it templating. Okay, clear? Any question, any doubt?
How this templating will be done in case of NGS?
So these are done using different reactions, enzymatic reactions, and also most of the NGS now uses robotics in doing that. So it has thermocycling, and also there will be selective enrichment of the libraries.
In our case, the process that we're talking about right now will be the same thing.
Yeah. So we'll also do the same thing. So I will show you in the. So I will show you in the successive slides. So how templating is done. So this is basically amplification and enrichment.
So we will amplify
Only the amplified part of 16S.
we will not fragment the DNA. Rather we will amplify.
So I will show you, because library prep has different strategies. So when we talk about WGS or transcriptomics, or so it has fragmentation step. So the entire genome is fragmented. But in case of targeted seed, so 16S is a targeted seed, or maybe some cancer patients. So they want to sequence. Okay, what are the oncogenes I'm having, or what are the mutations in the DNA repair genes, so tumor suppressors. So there are hundreds of genes. So we have to selectively target those genes. So in that case, the library will be prepared based on amplicons. So short primers are designed to cover all the loci. So basically we'll capture all the SNPs there. So no need of WGS. So these are different strategies of doing that. So for transcriptomics also, this fragmentation is necessary, but from RNA first we'll convert it to cDNA, and then we will follow the fragmentation steps. So these are different strategies of preparing libraries. But once the library is prepared, so you have to enrich the amplicons. So for that reason, templating is very much necessary. So why we enrich? To get better outputs, get better signal. Okay. And that part I will show you in later part. So what is targeted sequence? So I'm having different slides. I can just show you. So now, if we compare Sanger sequencing with the NGS, so this I'm comparing with computer because we can relate with our daily life. Now, when we talk about standard sequencing, so it's like old generation computers, mainframe computers. So we have heard about mainframe computers, mini computers, and personal computers, right? So mainframe computers are the very old generation computers. So these are less digital, more of kind analog instrument. So here lots of ancillary equipments are necessary. It requires a huge room. So and also the operation is very time consuming. So now in a single click you are getting all the result. But for operating mainframe you have to write pages after pages, then you will get some small output. So this we can compare with the Sanger sequencing. Now if you talk about the mini computer, so mini computer is upgradation from the mainframe, but here also you will require some manpower, some space. But yes, as compared to mainframe, mini computer is a bit easier for operations. So although this many large room is not required, but at least one small room is required and few manpowers are required. So one part will be taken care of one team, another part will be taken care of. So these are the initial NGS machines like pyro sequencer, then solid. So this kind of initial NGS platforms required because no robotics were there at that time. Automations were very less. Most of the steps were done manually, and then just to load the samples, just for reading the signal, all the operations are automated. This is for transcriptomic sequencing or whole genome sequencing, you will require a separate instrument. For targeted seq, you will require a separate instrument. For metagenomic. But now iTorrent can cater all the different types of sequencing strategies. And all the instruments, everything is totally automated, even the analysis pipeline. So here you will not require any bioinformatics tool. So you as a biologist can analyze the data. So it's just like plug and play. So you need to connect your equipment with the internet, you have to run the sample, and your data will be automatically analyzed using some clouds. So we have cloud-based apps or plugins, we call it plugins. So all these analysis pipelines are there with us. So just you need to upload your data, everything will be analyzed. And it can download, and it is a final cooked data. Even we have certain options. During the run, we can select those analysis options. So once the sequencing part is done, the machine will automatically give you the analyzed output.
What about privacy during cloud computing system?
So it is 100% private set because your profile will not be accessed by anybody.
It is protected by password.
Yeah.
Even not by any when you are from the site.
So it is totally encrypted. Even if you forget the password, gone. Even we cannot return. And also we have another options of standalone torrent server. So you can buy one server and you can locally do the analysis if you don't want any cloud, because sometimes what happens, there is the internet outage or maybe the network data speed is very less. So to avoid such things, and you have huge samples, so maybe today this is the start. After 1 or 2 years, Sagar may become the hub of entire Bhopal. So in that case, you might be running lots of samples from medicals, from other institutes. So in that case, you may require a standalone server. So the same thing. So, but don't think that if I have server, I need bioinformatics team. Not that. Your biologist can operate. So that's why I prefer to use this slide. So you can compare our technology with this personal computer. So here you will not require any kind of IT expertise or software expertise. So even a layman can very efficiently operate the PC. So this is what the NGS at the present is. It is now becoming more simpler, more user friendly. This is how the instrument looks like. So today, when we will go in the lab, you will see this instrument. So sir, I think it is S5 plus, right?
S5, only S5.
S5. So okay, so it
Yes,
is look,
this is.
yeah. So it will look like this. So this is a single instrument. So it has built-in server inside, and also there are fluidics which houses this chip. So everything is done with this single equipment. So there is no need for any ancillaries or any other big accessories. So instrument is sufficient to do transcriptomics, targeted seq, metagenomics, exome sequencing, you name it. So this is just a showcase of our technology. So I just wanted to showcase how the instrument looks like. Now we will speak about the semiconductor sequencing. So in most of the sequencing right now, the sequencers are more or less based on optics. So whether we talk about Illumina, we talk about pyro sequencing, we talk about Sanger sequencing, so everything is based on optics. So nanopore is still under development, so we will not discuss here. But this is something where we have replaced the optics with some sensors. So the same sensors that is used in your mobile phone or in the digital camera. So it is totally based on electronic sensors, and it is purely a chip based. So these chips are made from semiconductors. So this is an old picture. So when first iron torrent was devised, so this kind of chip were designed. So nowadays the looks of the chip is something different. But just let us take the example of the. So here in the middle part you are seeing some leaf-like structure, right? So now if you just enlarge it, you will see this kind of blocks. So we call it wafers. So this is like the same chip, same wafers that is present in any semiconductor chip, even in your CPU, computer CPU. So this kind of wafers are present. So this is the same kind of wafers. Now this chip, when if you examine very closely under a microscope, you will see this kind of groups. So what are those? These are wells. These groups are wells. So you have seen 8 well strip tubes. You have seen 96 well plate. So these are similar. So one well will house only one DNA fragment. So there are billions of wells. So billions of DNA fragments are loaded in one chip, and the entire fragments are parallel sequenced. And if you see this part, so you will see some electronic part, right? So these are the pH meters, which can sense the changes in the voltage or hydrogen ion. So these pH meters are most sensitive pH meter built by mankind. So it can sense the pH change of one hydrogen atom. So that's why the terminology iron torrent. So it detects on the basis of changes in the pH. So now tell me how this pH change happens. So I am talking about release of hydrogen atoms. So what is the relation between DNA sequencing and release of hydrogen atoms, hydrogen ions? Because this principle is applied in PCR, in Sanger also, but the detection principle is different. Even same thing is happening inside your body. So I'll give you some time. You think and tell me what is the relation between strand synthesis and release of H plus ion.
So when there is a nucleophilic attack and a phosphorestor bond is formed, so plus ions are released. Actually, ions are released. So changes.
Yeah. So H plus ions and 2-phosphate are released as pyrophosphate. So in pyrosequencing, this pyrophosphate was detected. So it was mixed with enzyme luciferase and then oxyluciferase reaction, so which gives some illumination, right? And that was captured by the pyrosequencer. But here H plus ions are sensed. So the pyrosequencing and iron torrent was developed by same person, Jonathan Rothbard.
the technology behind loading only one DNA fragment per visit?
So these are all dilution based. Yeah.
There is no possibility that there will be more than one fragment in the single. It's foolproof.
Yes, there will be like if you follow the protocol, if it is properly titrated, then around 10 to 20% polyclonality will be there, but 10 to 20% is very negligible. So if the polyclonality is say 20%, 25%, even 30%, so that is not a problem because that will be rejected as a noise. So what you have in your hand will be the sequence from monoclonal fragments. Means one fragment in one. So this is what is happening inside the chip. So when there is a strand synthesis, so the release of H plus ions is sensed by the chip and it gives some signals. So that's how the sequencing part is done in the ion tolerance sequencer, and this is the basis of identifying the sequence. Now there is another problem. If it is sensing only H plus ions, then how it can identify whether this is released from A or this is T or this is C or this is G? See, I'm asking you, why you are not asking me? Okay, so this is the detection principle. So I will show you how the machine can distinguish which signal is coming from the incorporation of which base. But this is the detection principle. This is the thwarting principle that is very much important. So you have understood how the detection is done. First of all, you understand how the sequencing is sensed. So this is how it sensed. So she told that when there is an elongation, when there is an incorporation of a BNT piece, then you please elaborate what she told. Yeah.
Sir, she just said that when it's elaborate and it breaks in H plus and P, 2P.
So from where it breaks?
Sir, the nucleotide bond when it breaks.
The OH,
Yeah, that one.
the hydroxyl group, there is hydroxyl group, right?
Yes, sir.
OH. So from the OH group, H is liberated,
Yeah.
and that O is used for formation of phosphodiester linkages, and the additional 2-phosphate 5 prime.
Yes.
2-phosphate will be released as pyrophosphate, right?
Yes.
So that H plus ions is sensed.
Yeah, that only you get
Yeah.
kilometer.
Exactly. So that is how the sequencing is sensed by the instrument. Now in the further slides, I will show you how it can differentiate whether the signal is coming from A or it is coming from C or it is coming from G.
So it may be changing color or something else.
No, there is no color added here. So what is happening? All the nucleotides are added not simultaneously, one by one. So in the first flow, only A will be added. Say for example.
Yes, sir.
Now if only A is added, then the other three will not be added at a time. So after the addition of A, this flushing of A, the sensor will see that whether there is any changes in the pH. Now if there is any changes in the pH in any of the wells, so that means what? That well contains the template containing T as a starting position.
Sir, it reflects more when it's come twice or third time out.
Yeah. The amplitude of the magnitude of voltage will be more if it is a trinucleotide repeat. So the pH change will be 3 times.
Yes, sir.
So if it is a tetra repeat, then it will be 4 times. But one problem is that if it exceeds 6, then there is a problem. So that is called homopolymer error in the release. So I think this is the same problem with the Sanger sequencing also. If there is a successive GG, I will be in a to say, dicey position, whether 5 Gs or 6 Gs, because the peak will not be separated properly, so it will be like this. That's very difficult to understand whether there are 5 Gs and 6 Gs. So similar problem is here. But that problem is not in the illumina, because it is going with the reversible chain termination reaction. So one part is elongated, means terminated, and then after the termination is done, again with the enzymatic treatment, this DNTP is removed and DNTP is added. Then again it is terminated. So termination, then normal extension, this takes place 2 and 4. So at least copolymers can be retrieved in that process. But one problem with the illumina is that it has a substitution error, high substitution error as compared to this. So to overcome the substitution error, the read depth is increased. So each system has its own merits and demerits.
So the DNA that was structured by the really single stranded or double stranded?
So it is single stranded.
Single stranded. So that means after isolation of DNA, it has to be denatured.
So there are steps of denaturation. So I
Because
was
the DNA that will
it will be double stranded.
It will be double stranded.
Double stranded.
And the one that is carrying is single stranded.
So actually when the DNA is placed here, so it is attached to some beads. It's called ionosphere. And here also we will add all the reagents for PCR. Here denaturation, ending extraction, everything takes place inside.
So initially it has to be denatured.
Yeah.
It's like PCR reaction.
So I'll show you, we have all these pictures. So this is the workflow. So behind all these things, this is simple workflow. First you prepare the library, and then you prepare the template. Templating is nothing but enrichment of the library, and then you just load the chain machine. Okay. So this is the workflow of technology. So most of our chemistry is based on MPC. We just so what is unamplified? So these are the platforms which is developed to capture the targeted seed. So mostly it generates short primers. Okay, so it will amplify the multiple targets. So don't think that only 5 targets or 6 targets. It can be 200 targets. It can be 2,000 targets. So this is a stage. But after this stage, you have certain other ancillary consumables like adapter. So all the fragments, whether we do amplicons here, generate thousands of amplicons, or you do the fragmentation of whole genome, the important part is that the entire end should be connected to some adapters. Why adapters are necessary? See, all the fragments have different sequence. Then in the subsequent step, how we can amplify? You will not have that many primers with you. So that's why some adapters are added. So adapters are known sequences. So it is universally attached in one of the ends. And there is another adapter, we call it barcode adapter. So what is the function of barcode adapter? Here also we will use barcodes. So what is the function of barcode adapters? Say for example, I want to sequence all of your sequence, right? So all of your sample can be loaded in a single sheet. And after running, how will you identify that which sequence corresponds to which person? मैंने आपका भी डीएनए लोड किया, उनका भी किया, सबका किया. अभी सीक्वेंस आने के बाद कैसे पता चलेगा ये सीक्वेंस बिलोंग्स टू यू, दैट सीक्वेंस बिलोंग्स टू हिम? So the sample has to be tagged with barcode. So the barcode is sample specific. So we have 16 barcodes stacked, we have 32 barcodes stacked, we have 96 barcodes stacked. So when we talk about for barcode number 1 to 32. So that means what 32 samples can be multiplexed. So that 32 barcodes have unique sequences. So barcode number 1 is unique, barcode number 2 is unique, barcode number 3 is unique. So all the barcodes are unique, and the software knows what is the sequence of barcode 1, what is the sequence of barcode 2. So barcodes are used to tag the samples. That's it. So in the Sanger sequencing, you are loading one sample in one wells, and you are naming the wells. Sample number X, sample number Y. But in the NGS, everything is pulled and loaded in the chip. Now, how will you differentiate which sequence corresponds to which sample? So that is being done by the barcodes.
But how are you going to do that? Suppose if you're doing microbiome analysis, you have purified or isolated DNA from any sample, maybe say intestinal sample or any soil sample. So that is having the DNA from different microbes. So how are you going to do this barcoding?
So actually, that is not this specific barcoding. So say for example, I have one sample, it is isolated from intestine. I have another sample, it is isolated from soil. So I will take DNA from soil, this is barcode 1, I will take DNA from intestine, this is barcode 2.
So even if I am tagging DNA from intestine, so that DNA will be having that DNA is from different microbes.
Yeah, so those microbes will be segregated.
Millions of microbes are
Yeah,
there.
yeah,
I don't
so-
know which microbes are there in the intestine.
No, all the microbes corresponds to intestine will have the barcode 1.
So all the microbes.
Yeah. So
Just
barcode is
for-
sample specific, just for tagging the samples.
Just for software identification.
Software as well as your identification, identification tagging, sample tagging. So
So if
separate.
you are simultaneously performing NGS for say intestine sample, soil sample, or some other sample.
So I can tag with different barcodes. So say for example, I have 100 bacteria from intestine. So those 100 bacteria will have barcode 1. Soil sample may have thousands of bacteria. So all these 1000 bacteria will have barcode number 2.
It's
So that's
something like lot.
yeah.
Lot. This
Yeah.
lot is
Just
from intestine. This lot is from
Yeah,
soil.
Yeah.
It's something like
Yeah. So it is
That-
basically used for multiplexing. So you can multiplex the cell sample.
The one thing I couldn't get you over there, you're between DNA or cDNA. What is the concept behind cDNA? Normally we understand that okay, you're having DNA from DNA, you're having a complete.
No, this is
complement
cDNA
DNA.
for
RNA.
this is for RNA-seq. So we mostly follow the implicit library prep here. So what is implicit? So either you can fragment the DNA or you design a small panels of primers. So after that, these steps are common. So that is being developed by implicit. So if it is transcriptome or if it is RNA-seq, you have to convert to cDNA. Or if it is genomics DNA, or maybe you are doing targeted resequencing. So targeted resequencing may have a small amplicons corresponds to thousands of loci, maybe 450 genes. So for example, we have comprehensive cancer panel. So it contains around 50 hotspot gene and the loci are more than 100. So similarly, we have carrier seq. So where prenatal screening can be done, and also for the couples. So before getting married or before having child, they can have their own sequence to identify whether their upcoming child is susceptible for any genetic disorder or not. So it contains 420 genes and loci are more than 17,000, 18,000. So all these genes you have identified. So the next part is that you have to ligate with some adapters, so that in the subsequent enrichment step, all the positive amplicons are uniformly amplified.
How foolproof is the prenatal screening? Because in case of eggs it's okay, but as far as sperms are concerned, during meiosis they undergo recombination, and even at the same time all those sperms that are released they are having different properties. So how effective is the prenatal screening? I don't think so. Because all the properties
These are all
the
germ line.
properties of genomic DNA is different, and the sperms that are going to fertilize it, its property will be different.
Not that, because see, when the samples are identified, so those target are there, so which are inherited, which are mostly germline kind of the genetic variants.
because somatic is fine.
Somatic is fine.
you can
So-
do, but as far as jump cells
So
are concerned,
those markers are all inherited markers. So whether you go with the sperm or we go with the
the same in the condition. The property for all the response are different. Because recombination is
so what this sperm or the egg cell or maybe so these are all tested on the basis of germline variant. So those genes are germline. Germline means what? It will be directly inherited, and also the sperms are also randomly checked. So if there is any such cases that the germline and sperms, these genetic aberrations or any kind of say copy number variations, so these are 100% matched, then the doctors can opt for abort the fetus. So these kind of applications are there. So whatever may be the applications, whatever may be your samples. So the next part is that you have to ligate the amplicons or fragments with some adapters so that it can be amplified uniformly in the subsequent step. Now, once the amplicons are developed, once the enrichment is done, then these adapters are again used for taking your sample in ionosphere. So that is something I wanted to show you. So these are the small beads. So it has a sequence complement. So these are the short sequences in the ionosphere beads. So these are complementary to the adapter sequence. So here your library has this adapter. Okay. So these adapters are complementary, and your library molecules can easily attach with the beads. Understood? This part is called the start of templating. So there will be certain beads, ionosphere particles, because these beads will be placed in the chip. So there will be certain scenario that the beads are empty. There will be certain scenarios where the beads are ligated with library, but some library may not have barcode adapters attached. So in that case, only this part will be attached, but the amplification will start from this part. So if the barcode is the P1 part is not there, then the amplification will not start. So P1 adapter is certain stretches of sequence that is attached with the barcode library. So that is again universal with the sequencing primer. So now somehow the barcode library, barcode adapters are not attached, and this part is not there, P1 adapter is not there. If P1 adapter is not there, then the actual sequencing reaction will not start.
suppose if you are doing the sequencing from only one sample, say for example microanalysis from soil sample. So I think in that case there is no requirement of barcoding because you are doing from only one sample only. Suppose say I am doing number of cell sequencing, but my sample is same.
But why? My question is why? Because you are using one chip. So one chip, using one factory chip, you can do 15 samples, 10 samples. So why you are using only one sample? So you are wasting the chip.
And every time you have to purchase the beans also.
Yeah. So instead of that, you multiplex.
The multiplexing is cheaper actually.
Cheaper. And also, sir, another thing that will also increase your confidence of the data. So that is applicable in any sequencing platform. Even with only one direction, one sequence, the sequence analysis is not authentic. But if you have multiple sequence of the same target or same region, that will give you more bigger picture, more comparative data you can present. So multiplexing is necessary. So the amplitudes will not amplify that. Even the frequency without barcode will not amplify. And if it is amplified, so then these are only few copies like that, or it will be like this. Denatured, but amplification has not occurred because one end is present and the other end is not present. So you require both the primer for amplification. Only this part will amplify where the library has both P1 and X adapter.
Both primer will be there,
Yeah,
and one
yeah.
gate will be.
So that is called enriched amplifiable library.
So what are the components during PCR? Because in normal PCR we have tag polymers,
So the same thing here also.
We'll be adding tag polymers,
Tag polymer is buffered, and in addition to that you have this immersion, so, and this kind of bits. So for enrichment, so these bits immersion takes place. So there is oil and kind of detergent immersion takes place. So there are billions of small droplets generated. So each droplet has all the components: bits, enzyme, DNA molecules. So now this become one reaction tube.
This thermo is designed like
There won't be any primers.
Yes, the sequence part means this amplification.
That is
primer-
from the
Yeah.
on the bits. So there will be separate primers.
Yeah. So that is not part of one master mix.
So in master mix,
Yeah,
all
it
the
will
components
have all this.
beads will
along
have
with
everything.
bits. We are adding bits and we are not adding forward and reverse primers.
So it has. So
It
these
has
bits
got
are
that.
called ionosphere particles, ISPs. So after this part is done, so you have to enrich. We need only this part. We don't want these, those, or any empty ISPs, because that will unnecessarily occupy your chip. Right? So how it is done? So these bits are treated with streptavidin bits. So this end is having biotin. So those primers are designed in such a way that the end part is having biotin level. And when the enrichment is done, this is done with the help of streptavidin. Now streptavidin and biotin, these are like husband and wife. They have good affinity. And this streptavidin beads is having a paramagnetic core. So just like iron. So it gets attached or attract towards magnet. So using this magnetic property, so there is a magnet attached. So there are certain attachment which contains strong magnets. So those bits will be captured by the magnet. So whatever things are pulled, it is actually pulling the enriched ionosphere. So the ionosphere with the empty library will be washed out. So whatever things you are having the actual amplifiable library. Now after this is done, so earlier it was done manually with the help of ES system. Now everything is done automated. So once this is enriched, now this part is loaded in the chip for sequencing.
Okay, so this all done in ion cell system.
Yes. So everything is done, means
Yeah.
from this step to this step, and even chip loading.
It's all done here.
Yes. So there are different components. So there are consumables. So these are the plastic parts. These are the thermal cycler where immersion takes place. Enrichment takes place. From here the immersion will be centrifuged to collect all these ionosphere particles. Now once the ionosphere particles is collected, it will be magnetically separated here. So once everything is done, it will be loaded in the chip.
So here we have to supply only the DNA. Rest of the thing will be done by this automatically done.
Not DNA means library.
Library.
Now for certain applications, the library can also be prepared here. But most of the thing library has to be prepared. It is better to prepare manually because you should have some controls, means how many samples you have to multiplex or so that this is nothing prepared, is just series of PCRs. That's all. So after that everything is automated. Plating, chip loading, everything is automated here. So once the chip is loaded, so here in the packet there are 2 chips. So with one consumable load you can sequence 2 chips.
Not use only one chip. to use 2 chips altogether.
It's better. So that is the way how NGS are run. So people wait for the sample. So they wait for sample collection. So once you have a considerable amount of sample, then you plan like that. Otherwise, what happens? If you have sample, for example, 5 samples, and now you wanted to run this 5 samples, you can run, but one consumables will be wasted.
What will be the approximate cost other than the chip? No, it can.
Because we take care from application R&D, that's it. Now if you ask about pricing,
I am asking because suppose rather than in place of two chip, if somebody is using one chip. So if it is in the range of some few thousands, then it's okay. But if the difference is very vast.
I mean, can't we ask this chip? No, it's difficult.
it's very difficult
Because I saw some video and people in France they tried to wash it and they were using it.
but we cannot guarantee that. But because what happens, they have more sophisticated implementing system, washing system. In India, we don't have, even you cannot purchase in any. So in that case, now you want manually clean. But say for example, it is not clean properly, then there is no noise. And trust me, if you consider sample extraction, DNA extraction kit, PCR, library kit, enrichment kit, everything, so that all becomes more costly.
Yeah, of course. I mean, one run is going to take.
So better if you can use more number of samples, sequence more number of samples. So that is more.
So how much sample you can load up at one chip? It's
Like in 530 for meta means at least 14 to 15 samples.
this time we are going to have metagenomics and we have more than 40 samples. This time.
So too cheap.
There won't be any waste this time.
Excuse me. So what's the reason for affinity of ISP beads and our DNA fragments?
So, as I told you, now this is the ISP bits, right? So in the ISP bits, you can see some projections, finger-like projections. So these are certain oligo sequence, and this is here after the adapter ligation. So the P1 and barcode. So this side barcode and P1 adapter ligates. This part has X1 adapter. Now this X adapter is complementary with this ISPs. So that's how your library molecules get attached with the ISPs. And when it only amplifies, the end part is biting level. The primers are biting level. So once you have the actual actual enriched template, so that has affinity with streptobidine, because streptobidine and biotin it's just like male female, means they have attraction. So this streptobidine beads has magnetic properties. So when you apply any magnetic field, these streptobidine beads are pulled. But when streptobidine beads are pulled, it is not coming alone. It will also take the enriched ISPs with it. So that's how you enrich the positive amplifiable libraries with you. And these beads are injected in the chip. And when this chip is loaded, so now everything is automated. But in earlier days, when 42 system was there, so it is kind of manual system. So during chip loading, we used to give sequencing primer, sequencing polymerase. So inside the chip, another round of sequencing takes place. So it's kind of sequence by synthesis process. So kind of PCR will take place. So here what happened? This strand synthesis goes on, and with incorporation of each DNT piece, this hydrogen ion releases record. So this is the automated system. You will see this we call ion shaft. bawarchi. So it prepares the template. It cooks the template for you. Okay. So in this direction the sequence takes place. Now each bit sits in the wells. Now I told you how the instrument senses whether the signal is coming from A or it is coming from G. So the sequencer is also installed with ATGC, means DNTPs mixed. Now the DNTPs are mixed in 4 different chambers. So we call it sequence cartridge. So you will see one box kind of cartridge. So it has 4 partitions. So one will have A, one will have C, one will have T. So all the 4 DNTPs will not be injected at a time. So it will be injected flow wise. One flow maybe T, another flow A, another flow G, another flow C. Now once a particular nucleotide is flowed, again it is washed, then second nucleotide will be flowed. So this is called flow cycle. So here actually what I have noticed, the orientation is little bit changed. So just I wanted to show you. So first, so if there is mismatch, it goes away. Now here there is some problem with the resolution, so it has just shifted. So the pairing will be done based on complementary sequence. So if there is C, then only G will pair. If there is A, then T will pair. So one by one the nucleotides are injected, and if there is any pairing, then only you will get the signal. So this is how you get the so if there is mismatch, it is washed out. So it is washed out. So this is how the entire sequence is read, and this is the voltage part. We call it ionograph. So you will get this kind of low voltage peak. So you will not see it. The software is going to convert it to ATGC signal. But after the run is over, we can show you all these raw attributes are also present. So this is converted to reads with the help of base calling software. So if there is any means homopolymer, so one of you was asking me if there is 2 incorporations, so the intensity of the graph will be doubled. So looking at that P, we can identify whether this is homopolymer or this is means dinucleotide repeat. It is trinucleotide repeat. So after that you will get the entire sequence. This is how the instrument looks like inside of the equipment. So these are the different chips. So depending on the output, what is the read length, what is the output you require, you can use different chips. So one instrument is sufficient to use multiple types of chips. So this is the advantage of our S5 sequencer.
So right now we're using 510,
530.
530.
We'll be using 530.
But we need for 50 to 20 billion reads.
Yeah. So that is sufficient for metagenomics and also small genome.
So we're going to use this 2 to 3 million reads.
So these are very low, means if you have very lesser sample size, and also it's very small genes, say only BRCA you want to sequence.
Certain mutations.
Yeah.
Certain mutations and all.
So this machine is actually very versatile. It means it is a medium throughput instrument, but you can use it like a personal machine. And also it is used with the diagnostics. So you have the flexibility in the run protocol. So very small medium experiment can be done. So that's why one instrument can cater all types of.
Thermophotic is when you want to get comprehensive
Yeah. So see, for internet we have different. So just let me complete this part. So these are the consumables part. So here the DLTPs are there. So just you have to put the cartridge inside the instrument. So these are the cleaning solutions. So this is just comparison. So how is the time duration of one chip? So if you are using 200 base pair chemistry, now if it is 5 times chip, it can take 0.5 hours. So like that. So with the increasing time or of the chip capacity, so these are the different run time. And also with the different instrument model. Now if you are using S5 Prime, it is most fast machines. But S5 is the basic. So it is much more fitted for academia. But S5 Plus for medical colleges is good. S5 Prime is very suited for diagnostics. So depending on the speed, depending on the capacity. So I'll not go to this part. So
in our case, it will take approximately 2 hours.
2 hours for completion of the run and assembly it will take. So what you can do, you can just wait for few hours. So you will get the assembled done.
How many slides do you have?
So these are the different clinical applications we have. Means, oncology research, inherited disease, microbiology. So we have certain panels with us. So I'll not discuss it here. So we have comprehensive different panels. So panels means all the known genes are covered here related to different cancer types. So for reproductive health also we have NIPT solution. Now if there is any prenatal testing or any newborn screening, postnatal testing, so all the solutions we have with us. And for infectious disease we have this microbiome kit. And also for metagenomics we have microbiome research kit. So today we will go with the metagenomics kit. These are the certain softwares we have, a basic designer. Now you want to custom made your panel. So maybe you have, say for example, somebody is doing some medical research. Now you have identified 100 genes or 200 genes associated with certain disease. So you can build your own panel here. So instead of buying, you can design your own panel. So there are designer tools available. Just you need to upload your template sequence or gene of interest.
So these are free software?
Yeah. Just you have to have account in Taiwan.
I mean, if I want to give this to my students, they can also use it.
So this is called implicit design. So you can also opt out validated design panels, and you can also have your own custom made panels. And these are torrent suits. So I will show you torrent suit because it is very much necessary when we operate the actual instrument. So run plan, you can monitor the run, what is the run progress, how many times you are remaining, and what is the primary data you are getting after the run. So all these can be accessed through torrent soup software. And another important software is Iron Reporter. So that will be shown by Praveen. So here all these analysis plugins, apps are present. So it's just plug and play instrument, nothing is required, no bioinformatics specialization is required yet. So this Iron Reporter is again connected to another databases, IDB. So you will hear about IDB when you do the human gene research or human gene sequencing. So here all the sequence will be assembled, just like multiple sequence alignment. And from there you can get to know that what is the alternate alleles, what is the recessive alleles, or what is the dominant alleles. So IGB is a very beautiful software. So if time permits, it can show you. So how it looks like. So I'll not discuss anything regarding bioinformatics part, that will be taken care by Prabhu. So this is not important. So any key questions you have? Anything you want to know?
Any question from your side?
Yeah, please.
Sir, in the case of infested DNA sequences, like diseases which are coming nowadays, coronavirus, which keeps on transforming their protein, is it this machine is capable of
Yes,
finding
yes.
accurate nucleotide
Yes,
bases of
yes,
the DNA
yes.
sequences.
Yes, accuracy is high. We have lots of publications for COVID sequencing also. So we have COVID genome sequencing applications with us. So I have not shown all the slides here. So it requires another session for me to show. So COVID sequencing can be done, even like WGS protocol can be used here. So that after the DNA or RNA is extracted, you click and then adapt a ligase. So it is tested here. So this machine is used for diagnostics.
Actually, one of our participants has got an insect. And he is saying that insect is biting somebody and it's causing some disease. Suppose if you want to find out anything with respect to those which microbe is causing that particular disease, what can be the approach?
So first of all, are you sure that it is caused by any microbes? Means there could be some lesions or some injuries. So first of all, is there any sign of infection? Is a microbial infection?
I think I just showed you the images.
Suppose if there are some blisters or something, it can be due to allergic reaction or
Allergic,
due to
no,
antibody.
no,